Good morning, good afternoon, and good evening to all of our webinar participants, and welcome to the first webinar in our series on microplastics and health. My name is Daryl Boverhoff, and I'm a Sustainability Director with Dow, and I'm one of the planning committee members for this webinar series. Full information about the series, including planning committee roster, can be found on our website. This series is an activity of the Environmental Health Matters Initiative, a forum of the National Academies, for which I was a member for the last several years. The intent of this series of webinars is to provide a solid baseline of information and knowledge to inform future work and decision-making on microplastics. More specifically, the series aims to assist in identifying research gaps, regulatory needs, and really promote cross-sector dialogue on decision-making regarding microplastics and their potential impacts on human health. So, on behalf of the Environmental Health Matters Initiative and in collaboration with the National Academies Roundtable on Plastics, I'd like to welcome you to our first webinar in this series that's going to focus on methodologies for appropriate and improved sampling, analysis, characterization, monitoring, and reporting of microplastics. So a lot. So why start with a webinar on methods for sampling and characterization? Because this is a foundational capability to support exposure assessment and hazard evaluation as we seek to assess potential human health risks. The goal of this first webinar is to promote awareness and provide standard baseline understanding on the importance of sampling and characterization, and future webinars are going to build on this as we address topics around distribution in the environment and human exposure pathways, potential hazards to human health, and the complexities of assessing risk, and considerations for risk communication and exposure mitigation. That's a lot to cover in this series, but we are confident that this will provide an effective baseline of information to inform future research and discussions. I'd like to emphasize that the content provided in this webinar is not intended to represent the formal perspectives or recommendations of the National Academies. All views, opinions expressed are solely those of our volunteer guest contributors. This workshop series aims to bring together a variety of stakeholders and experts from different sectors with diverse expertise and experiences. A goal is to foster scientific dialogue and collaboration through productive yet balanced discussions. For this workshop, we want to thank the Planning Committee and members from the Roundtable on Plastics for their thoughtful contributions, the National Academy staff, as well as today's speakers and panelists, all of whom have worked hard to bring together this exciting event. Now, let's turn to today's webinar. We're going to have two sections in this webinar. The first will be a coordinated presentation to set the stage, followed by a short Q&A. We will then turn to our second section, which will be a multi-sector panel discussion that will delve a bit deeper into some specific topics and questions. Audience members, you are invited to submit your questions at any time using the Slido, where everyone can also upvote the questions that they want to see answered. The link for the Slido can be found on our website and in the chat. So with that, we're going to get started. Again, the goal of this first session is to highlight the best practices and considerations for the collection, measurement, characterization of microplastics in different media, explore the latest scientific findings that could enhance the comparability, reproducibility, and transparency of microplastics research. For our first session, it's my pleasure to introduce our two esteemed speakers, Dr. Jean Hankett, who is the Sustainability Lead for the Corporate Analytical Material Science Department at the BASF Corporation, and Dr. Amari Walker-Franklin, who is a research chemist at RTI International and a member of the Roundtable on Plastics at the National Academies. All of the biographies for our speakers and participants can be found in the agenda on our website. They will have 25 minutes to present their thoughts, and then we'll open up the session to questions. Again, as a reminder to our audience members, please submit your questions at any time through the Slido. And with that, Jean and Amari, please unmute yourselves and start sharing your screen. Again, you'll have 25 minutes for your presentation. To keep us all on time, I'll send you a couple reminders, 5 and 2 minutes in the chat as we reach the end of your talk. With that said, I'll turn over the webinar to you.
All right, thank you, Daryl. So thanks again for the introduction. Jean and I were tasked with a very large endeavor to try and fit as much information into 25 minutes on, methodologies for microplastics analysis and characterization. So we're going to try and move relatively quick. And I think it's not sharing the full screen just yet, but the goal is really to provide you with as much information and then a lot of references that you can continue learning from. So please, once the webinar is over and the upload to this presentation is there, feel free to go back and look at some of these referenced links. And then, is it on slide 1? There we go. So the goal for this agenda is to introduce, microplastics, just do a reintroduction based off of the last webinar really quickly, and then discuss the current status and methodologies for microplastics characterization. We are then going to go over some of the opportunities for innovation and improvement in the field. And again, please note that these slides will be available at the end, and we really encourage you to take a look at those links. So next slide. So defining the terms. So we pulled these from the last presentation on the introduction to microplastics and human health implications. And Robert Ellis Hutchings noted from these slides that microplastics are defined as plastic particles that are smaller than 5 millimeters. And then microplastic, again, next for the animations to do the full slide. And then microplastic fibers are typically characterized as microplastics that are lengths between 3 nanometers and 15 millimeters with a length to diameter ratio of 3. And then with the assumption that anything smaller than that is what we would call a nanoplastic range. So for this purpose of the conversation, we're mostly talking about microplastic size ranges. Next slide. And then again, what are plastics? Well, plastics are defined as high relative molecular mass polymers. And then they have a variety of different repeating low mass molecules. And there's so many different kinds of polymers, nearing even the thousands of formulations at this point. But here are some of the most common polymers that you can see on the slide, including PE, PET, PVC. Next. Oh, one more. Oh, and then, the world of nanomaterial research has been incredibly informative for the microplastics field, as we learn that these particles are really long-lived and ubiquitous in so many environments. And so some of the sampling and extraction analysis techniques sometimes use very similar tools and techniques. So microplastics are incredibly unique because of their complexity. There are so many different polymer types. They can be in different layers of composites. They can also come in different shapes, like these spheres, fragments, and fibers. And they have different aspect ratios or even surface chemistries that impact, the addition of other contaminants potentially to the surface. In addition to just the polymer type, we also have a wide array of chemicals that can be incorporated into the polymer matrix that are not chemically bound, so they can then also diffuse from these matrices over time. In addition to the polymer surface is also sometimes changing depending on the environment that it's in, like, biofilms accumulating on the surface to UV weathering, increasing oxidation, or even hydrolysis from exposure to water. So these surfaces are changing quite a bit in some ways. And so it is, when we're thinking about acquisition and sampling and analysis of these microplastic particles, it's important to try and recover all the particles, but we also need to do some manipulation so that we can properly identify the polymer type without destroying the microplastic itself. So we have a very complex challenge in itself. Next slide. So to get us started on our journey towards, what does it take to gather microplastics, we have to look at these 3 steps of microplastic characterization via sampling, extraction, and then analysis. So each of these steps are very important because loss and or contamination can be introduced at many of these points. And additionally, the way that we're preparing and analyzing can give us different kinds of information about the particles. So it's really important when we're thinking about the reporting standpoint to try and get a lot of information on particle number and particle weight when we're thinking about hazard and risk assessments. And working to use chemical-based identification methods so that we can avoid false positives and false negatives, which is really critical. Next slide. Oh, so we're going to first start with sampling. Next slide. So the research is really rapidly expanding to study the prevalence of microplastics in environmental samples like air, water, plants, compost, and sediments, and then into the world of biological samples now with human and animal tissues, cells, and I recently saw a paper on bones, microplastics getting into different cartilage tissues. So at this stage, there's not as many standardized methods for sampling in these compartments, specifically for microplastics, but the methods that we're linking below are some of that have started in these realms, especially for water. And then there's also this ISO standard that provides some general guidance for sampling. So next slide. Water analysis in particular has been one of the more widely studied for plastic pollution in general. Generally, most collection trends towards using nets or, Newston, manta nets, plankton nets, or even sieves. Though some of those issues with these is that there could be clogging and also sometimes sample volume challenges. And other analysis sometimes will also grab some of that water sample to analyze later, which is a little bit more labor intensive and can be costly. However, when we're also looking at the fact that most of these studies are looking at mainly environmental samples like freshwater and marine environments, but we're now starting to see in the research that there's an increase in drinking water analysis, especially with some of the new regulations from California to study and potentially monitor for microplastics in drinking water. So this new ASTM method, which is the standard practice for collection of water samples in variety suspended solids, is a great start towards standardizing collection practices for these water sample types. And you can see an image from one of the papers of an example pump system that goes through a series of filters to try and isolate these microplastics and controls for the sample volume, because you tend to need sometimes thousands of liters to get an accurate representation of microplastics in these environments. Again, the challenge from these methods is that we're still losing a wide variety of microplastics depending on the opening. So some are less than 330, so anything above 330 is what's captured, but the smallest is down to 20 microns. So anything smaller than 20 microns for microplastics is not always going to be retained because of these limitations. Next slide. So in the collection of air studies, there are, quite a bit of, unique technologies or techniques that can be used based off of the industrial hygiene standards like OSHA and NIOSH. So there's, generally a good start for where we can go and start to collect using things like filter cassettes, cyclones, particle impactors, are really great for trying to collect things like PM10 or PM2.5, which are the most relevant for human health. But if you're trying to look at something that's also microplastic prevalence and transport that are including particles larger sometimes than 10, using things like wet and dry collector buckets have been shown to also replicate or represent regional global transport of some of those larger microplastics too. So it's really important to try and look at some of these opportunities for expansion. Next slide. And then within soil, sediment, and compost. So generally, sediment's also been another area that's been sampled quite a bit. There's quite a few different recommendations from different groups. Some say that you need quite a large quantity of sediment to be representative. So NOAA recommends 400 grams of wet weight per replicate, which is quite a lot. And MFSD recommends at least 5 replicates within the top 5 centimeters of every 100-meter block. And when you're talking about how to sample, usually things are techniques to use box cores or grab samplers. For soil, there's not as many standard methods, but for compost, this new ISO standard is in the works. It's still being developed, but it's on its way to discuss methods for compost extraction, because it's such a homogeneous sample in comparison to some of the other sample types. Well, it's such a heterogeneous sample that you have to try and make sure that you have a good mixture for analysis. And then finally, for human health, we can see in the last 20 years the sheer increase in number of papers that are gathering attention for microplastic analysis and isolation. But it really falls under two camps. One is utilizing these methods for controlled toxicology studies and then being able to sample for them at the end. So usually the field is currently using more standardized polystyrene commercial beads or fluorescently tagged particles to try and look at transport, but not as many of like the environmentally relevant types of particles. And then the second camp is looking at exploration of biological samples, which is a different challenge because there aren't really many standard methods, and when they're sampling, it's usually can be in a high contamination risk environment sometimes. So there are some of the newer studies that are trying to reduce those contaminations, such as using plastic-free samplers, glass blood tubes, metal bins, to try and reduce some of that contamination at the beginning. Next.
Thanks, Amari. So now we're going to dive into our second section, which is extraction. And this is where we're really going to start diving deep into the science. Now, Amari's mentioned the phrase matrix, and we're going to continuously talk about this today. So what do we mean when we talk about matrix and microplastics extraction? Well, when you first think about microplastics in the environment, perhaps your idea may be straightforward. We have larger plastic pieces that eventually go to single molecules, but we found over the years that simply isn't the case. We have a lot of interactions with biological biofilms, oxidative mechanical breakdown, aggregation, and all of these materials, interaction with the environment, that's what's considered matrix. So when you extract your microplastics, you can't chemically identify them or properly physically identify them without removing that sample matrix. Now, your microplastics should be prepared considering the type of matrix, your method of analysis, resulting potential for false positives or negatives, and then the chemistry of the microplastics themselves, because the chemistry can be impacted by your method of extraction. So ideally, your matrix removal should be reproducible. It should allow microplastics to remain intact with high percent recoveries and low signal interference. But this is a great time to step aside and talk about quality levels for microplastics in general, microplastic science in general. And in-depth literature reviews indicate critical needs for quality development of the following areas associated with our three parts: sampling, extraction, and analysis. Those four areas for improvement are contamination prevention, blank protocols, additional quality assurance and quality control, and then the need for improved reference and standard materials. So the reality is, as more complex samples are analyzed, as Amari noted, we started with water, and now we're looking at these more complex, soil, compost, biological samples. As they increasingly become more complicated, data interpretation is becoming increasingly complicated. So I'll talk about this a little bit in the analytics section, but for now, we'll quote an excerpt from the 2022 World Health Organization report on dietary and inhalation exposure, stating the available data are of only limited use for assessing the risk of nano and microplastics to human health, partially due to the use of bespoke methods. So how do we ensure that there's high quality associated with the extraction of microplastics and analytics and throughout the whole process in general? Well, let's talk about blank protocols and QA/QC first. So you may or may not be familiar with the concept of blank protocols. It's effectively trying to get an understanding of what could be that potential background contamination. So whether you're in the field where you want to understand not necessarily that compartment of interest that you're sampling, but right next to you, let's collect, see if there's any microfiber contamination. We would want to do this for experiments and run those blanks, but we also have lab or process blanks. So when you're working up your sample, you're extracting it and you're analyzing it, understanding in the environment that surrounds you what are the potential for contamination. And then there's even a third type of blank known as procedure blanks. Procedure blanks is where you run your experimental extraction with no microplastics present to understand if there are potential false positive or false negative effects from that matrix that may or may not be remaining at the end of your extraction. And then lastly, while you're running these blanks and the procedure in the laboratory for extraction, you want to understand the chemical effect. So if you're oxidizing your material, you'd run an oxidation on reference materials that are of the same chemistry of interest. So if you're running polypropylene, do your oxidation on reference polypropylene materials to understand how that impacts your analytical results. And then lastly, it's often useful to better understand the environmental impacts to do what you can in the laboratory to mimic any chemical or adverse changes to your microplastics while they're in the environment. There's no way to fully mimic this, but we do what we can in the laboratory. So related to the concept of blank protocols is contamination prevention. And there are a variety of different items listed here that are generally well accepted best practices for reducing contamination. But I will say today, the number one best practice is method transparency. So researchers sharing back and forth what they have done and what they haven't done to prevent contamination. That's really important when it comes to then understanding what the results mean at the end of the day or at the end of experiments. And then when we talk about our fourth category, quality assurance and quality control, my general recommendation is to consider ISO quality principles. And so you may or may not be familiar with ISO quality lab programs such as 9001 or 17025, but the concept is that they provide transparency for the laboratory and general guidelines to improve the quality of results. We can take some principles from these ISO quality programs, like conducting percent recoveries for your extraction, otherwise known as environmental spikes, which should be conducted for relevant particle sizes, particle chemistries, and concentrations. Then there are things like control standards, which can be run against calibration curves. You want to run all your different blanks when you're analyzing your samples from the environment as well. And then again, you want to understand what are the chemical and environmental impacts on reference materials, and most important of all, evaluation of the impact of the matrix for false positive or false negative potential results. And so given that we're getting into more complicated areas, we really need to make sure that the appropriate reference standards are analyzed alongside samples. And so I'll hand it off to Amari to talk about this area.
Yeah, just really quickly, because I want to make sure there's some time for analysis. But there are some unique differences between a reference material, which is manufactured according to strict specifications, like certified NIST SRMs, to standard materials that are often analyzed and certified with traceability that could be used for things like instrument calibration. And the need for references and standards will differ between if you're using it for a toxicology study or if you're using it to test like recoveries for environmental standards. So we need a vast array of them. And here are some of the ones that are linked currently that are currently available, but this is definitely a gap that needs to continue to be expanded upon. And then the other idea for references include reference libraries. So here are a few example reference libraries for Ramon and FTIR that we're going to talk about soon. And then for sample preparation in particular, when we think about, how to prepare things like that for tissues and organs, it's really hard to remove those matrices. We've got a lot of fats, oils, lipids that can get in the way of the analysis. And while there's no standard methods currently, there are some, common trends as far as utilizing a digestion step of some kind, whether that's using basic or acidic digestion or things like hydrogen peroxide, which have been seen more often. It is a challenge to really try and get full efficiency of removal. But these timescales for digestion can range sometimes from a couple of hours to days to weeks. And the digestion efficiency has very limited information right now from these method papers. Next slide. And then as far as sample preparation for other matrices, there's some existing standards for water, but for other matrices like soil or other materials, the most common, again, includes density separation of some kind. And, just to put a plug in for the NIST, And Hawaii Pacific University's device, they just built a plastic-free device for density separation that uses sodium polytungstate. And so one thing to note is that polymers all have different densities, and we have to use higher density gradients to be able to try to separate them in solution from that other organic material. So this is a great method for the start to try and remove as much material as possible and isolate the microplastics. And then we have to use things like oxidation, try and get the rest of the organic materials off. And then we'll go next.
All right. So now we'll continue on to part 3. So we've appropriately extracted our material, we've appropriately sampled it. How do we analyze it? And I'll just go over this at a high level today. We can discuss more with the panel as needed. But at minimum, when we're talking about analytics or characterizing microplastics, the microplastic study should have confirmed chemical identity and confirmed presence of solid particles. So there really are 2 different camps of analytics, which I won't go in depth into all these different ones today, but suffice it to say, we can determine how many microplastics there are in a sample versus what is the weight of those microplastics. And some of these analytics have chemical identification tied into understanding how many or what the weight is, and others do not. Lastly, if you want to further characterize your material, there's a wide suite of analytics that are appropriate for polymers. I did want to take a quick dive into some of the most popular count-based spectroscopic methods, and note that there are standard methods in development for microplastics originating from water for both IR and Raman. So Raman and micro-IR spectroscopic imaging remain the dominant methods for count. Expertise and experience is still required to appropriately understand the results that we're obtaining, but we have a lot of statistical tools and machine learning that supports analysis right now. There are both benefits and limitations for Raman, including differences in the theoretical lateral limit of detection, or how small you can see particles, what could interfere with the signals like water or dyes. And in both cases, there are false positives that are possible. So this includes oligomers, stearates, oils, fats, and surfactants, especially for polyolefins, if you're looking for those types of microplastics rather. And the general rule of thumb is you need to have reference material to match for identification. You should have a high percent match, at least 80% to relevant reference materials. But there's reasons why these are the most popular analytics, and that's because they are really powerful. We can see tens of microns or down to 1 micron for traditional Raman and IR microscopy. But these are often also complemented by mass-based methods, as I discussed before. And one of the most popular mass-based methods is called pyrolysis GC mass spec. Standard methods exist, again, for microplastics in water, but not for more complex matrices. And pyrolysis is a really interesting case here because it's an extremely powerful analytical technique. It gives you chemical specificity. There's an extremely low limit of detection, less than 0.05 milligrams as an example for quantitation. But we have a lot of false positives and negatives possible with this technique, and I'll tell you why right now. So effectively, when you conduct pyrolysis, what you're doing is you're heating a sample, really high heat, let's say 500 to 600 degrees Celsius. That then fragments your plastic into smaller molecules. Those molecules are separated on a column and detected by mass spec. But what you're detecting isn't necessarily those polymers directly. You're detecting those pyrolysates fragments. They generate these patterns, and you're actually having a lot of reactions occurring under pyrolysis. So if you take PVC, for example, you're not looking at the PVC molecule to detect PVC and quantify it. You kick off the chlorine under these conditions, and you're forming these aromatic rings. And that's what you need to find to determine that PVC is present. So what's really interesting then is for biological samples where you have high fats, lipids, you not only have a lot of interesting interactions that can happen at this high temperature, but you also have within areas of quantitation or qualitative identification of microplastics, potential overlap of signals from these naturally occurring materials. And so that makes it expertise an absolutely critical component of analysis for pyrolysis, even more than other traditional analytical techniques. Now, very briefly, I've discussed that these are complicated techniques. These are complicated extraction mechanisms, as Amari pointed out. And so historic interlaboratory studies tell us that reproducibility in results in even controlled conditions is difficult. I've listed 2 different international studies here, one with the Southern California Coastal Water Research Project and one in Europe. I won't go over the results today, but we'll say you can see there's a wide range in percent recoveries for microplastics in both of these studies. We've improved significantly, but there's a long way to go, right, in terms of growth for our 4 improvement areas. Now we know there'll be future advances, including lowering limit of detection. Standard orgs and government orgs will continue to develop methods and guidance, but it's a really complex topic. So no one group can improve the level of scientific knowledge alone. Increasing collaborations, information exchange is absolutely key. And a great example of that is SCORP in California. I'll take a second to talk about 2 outcomes from industry as well, MARI-E and Operation Clean Sweep, and how we can learn from those opportunities and industry efforts. So if you're not familiar with it, MARI-E is a Microplastics Advanced Research and Innovation Initiative. Here, it's run by the International Council of Chemical Associations. A variety of different organizations work within it to develop a holistic risk assessment framework for microplastics and nanoplastics, advancing analytical capabilities, hazard, and exposure. And then a shout out to Operation Clean Sweep here, which was initiated in the US in 1991. It's a global program for the prevention and containment of plastic resin, pellet, flakes, and powders. More than 5,000 companies are now participating, and it's a commitment to proactively implement good housekeeping practices. Why do I mention it today? And we're talking about research, because there's a lot of information sharing available. So manuals, best practices, reporting, third-party auditing, and certification. And then lastly, we didn't talk too much about next generation or cutting edge advances in analytical technologies. They're out there. This will continue to develop, continue to move us forward. We have some fantastic and interesting analytics, including microcavity size selection in Europe, nanoflow assays from NIST, hyperspectral imaging flow cytometry from the DOE and EPA, and of course, new discoveries are made every day. So with that, wrapping up our take-home message and acknowledgements, microplastics investigations began in aqueous environments, so unsurprisingly, the next frontier to tackle are these more complex matrices, so additional environmental compartments, biological samples. Advances in analytical technologies have allowed for improved analysis, but multiple challenges remain for false positives and negatives, contamination prevention, QA/QC, and reference and standard materials, and increasing levels of quality and transparency in research will be absolutely critical to aid in the improved understanding of exposure, hazard, and risk. New generation of standard methods, guidance, and referenced standard materials will support this. And of course, lessons and exchange between multiple sectors will further enhance breakthroughs in microplastics science. And with that, I'll hand it over back to Amari.
Oh, I think it's well said. And I think we're on to the next part.
Perfect. Jean, Amari, thank you so much. Great overview in a short period of time. Clearly a lot to cover. So I can hear everyone who's on the webinar clapping for you. So thanks so much for that great overview. Very systematic, very thoughtful, and sparks some questions in my mind. So my first question is, clearly this is a complex space. What advice would you give to a researcher, maybe a biologist, toxicologist, who wants to start assessing microplastics? What type of key partnerships should they form? I'm reminded of when I was in grad school and genomics became a big thing, and you had biologists that needed to partner with computational folks to create this whole area of bioinformatics. So what are some of the key collaborations that need to be formed to make sure that the data we're generating on microplastics is most useful? So Jean, we'll start with you.
Yeah, absolutely. And I think you hit the nail on the head already, Daryl. So having that cross collaboration in different STEM subjects is absolutely vital because you want to have an understanding of statistics, of physics, of chemistry, of toxicology. How does that relate to hazard and risk? Not just taking a sample and then having a direct correlation. So really support networking and digging into an understanding of how those different topics all interconnect.
Yeah, and I'll just add on that one, I think after all the work from the presentation, go through those slides, go through the links. I know it was really quick to look at all that information, but that it's a lot of helpful material to get started. I wish I had something like that when I started. But and then, from the other side is that there's a lot more conferences and meetings that are very microplastics themed. And that's a really great way to start building those connections. I know that even at Duke, my last conference, last university I was at, they have now the Duke Plastic Working Group that is just across all of these departments that are working together in trying to address this huge problem.
Perfect. So health professionals, toxicologists, make friends with analytical chemists, material science folks, right? And maybe go to these conferences or find them and form these collaborations that are going to result in really strong scientific research. Maybe one other quick question. We talked about the importance of controls, whether that be blanks or standard materials. How widely available are these standard materials, and where does one go to get these things? And what is the current development status on some of these?
Absolutely. So Mari, I'll be sure to leave some of this to you since you presented that slide. But from my perspective, Daryl, they're not as readily available as one might think. Now, part of the reason is because it's difficult to make microplastics within that size range of interest by mechanical abrasion, for example, or milling. It's hard to make microplastics, period. So that's one major, I would say, not roadblock, but speed bump is who's going to make these materials and how much energy it would take and who is also going to certify them. But that being said, there have been a variety of advances, and this is where I'll hand off to Amari to talk about some of the kits that have been developed for these simple, more model type of plastics.
Yeah, I think that there's, even from commercial lab standpoint, so it just kind of, I think I noted in the slide that if you're thinking about the toxicology side of things, sometimes you want a more homogeneous type of sample. So there are nanospheres, polystyrene is probably one of the more common ones. And then for the polymer kits, there's now a variety of I think up to 22 different polymer types, ranging in different sizes. So sometimes you still have to mill them down into smaller groups. And then I know that there's a lot of innovation that's still currently continuing across different universities. Even here at RTI, they're trying to produce their own nanoparticles or nanofibers using electrospray, electrospin technology. So again, another one of those collaborate across institutes and analytical chemists talking to toxicologists to get these things going. Because yeah, it's going to take a lot, not only just to make the particle, but then also make it environmentally relevant. Because you might also need to expose it to light or to water or, put it in soil, and then it looks more representable. So it's this long time in coming. It's going to take some time.
Perfect. Well, hey, what I heard is super complex, form collaborations, do good science by having the right controls and the analyses. So follow Amari's advice, go back, look at the slides. There's lots of resources in there to really help set that baseline in terms of how do we do good science to help with the assessments of microplastics. So with that, Jean and Amari, I want to thank you so much. We're going to now transition to session 2, which will be moderated by Dr. Rachel Meidel from our planning committee, and she's also a member of the Roundtable on Plastics at the National Academy. So Rachel, over to you.
Thank you for that introduction, Daryl, and welcome everybody. So what we're going to cover in this discussion is highlighting the successes in studying microplastics, as well as the lessons from past experiences across sectors that we can apply to future work. And we're also going to identify where progress is occurring and focus on the gaps that's going to allow us to build our next steps in exploring the connection between microplastics and health risks. So each of the new panelists joining us for this session will give a 2 to 3 minute opening remark to cover a brief overview of the background and some of their initial thoughts on where they see opportunities in understanding the complexities of microplastics. And this is going to be followed by questions from the audience to the full panel. So our first panelist is Dr. Alessio Gamiro, Senior Researcher at the Norwegian Research Center in Norway. So Alessio, if you could turn on your camera and unmute your speakers. And just a reminder to all the participants here that you can submit your questions to us following our remarks from our speakers. So Alessio, the floor is yours.
Thank you, Rachel, and good morning, everyone from Norway. So my reflection about the plastic research is like we have recent advances in the detection methods, have significantly improved our ability to identify plastic and microplastic across various environments. This progress is crucial for obtaining accurate data and understanding the extent of the problem. Furthermore, interdisciplinary collaboration among scientists, industry representatives, and policymakers have made it instrumental in the uncovering of sources and pathway of microplastic. Public awareness campaigns have successfully increased the understanding and prompted behavioral changes regarding the plastic use. Perhaps one lesson is the importance of standardizing research methods, as we discussed in the previous session, which facilitated the data comparison and enhanced the reliability of findings. If I'm mentioning a little bit what was my history about the plastic research that I did 20 years ago, at the beginning we had no clue at all about what plastic pollution was, no idea what was like the analytical method most convenient, most reliable, most performing in the characterization. We were not actually really aware that we were preparing the sample in the appropriate way. And once we sorted out what was the analytical method, then it was also complicated to understand where this plastic pollution was coming. So integrating a little bit more our knowledge with other expertise on a cross-disciplinary, interdisciplinary type of environment. So these are like examples that can be like brought, showing that we need interconnection across a different type of expertise. Additionally, the integration to the scientific research into policy development has proven vital for the effective action, strengthening the communication between research and policymakers. Again, regarding some area where we see some progress, we have a technological innovation that have led to the development of advanced tool for plastic sampling and analysis. You may imagine that at the beginning, we trying to target the plastic pollution were literally surrounded by plastic in our lab, in our boat, in our equipment. So there was, and there is still a lot of work that is done in order to have like equipment that is not actually contaminating our sample, for example. Some samples we are expecting to have a lot of plastic in, sediments, wastewater treatment plant sludge, and other type of material like this. We are expecting tens of thousands of plastic particles per gram, per kilo of sample. But when we are talking about drinking water, which was like I mentioned before, yes, in that specific case, there is the risk that during the collection of the sample, you may also be able to contaminate. And the same comes with the sample preparation prior to the analysis. So research is progressing and should progress more on this specific direction, with a specific eye on the possible contamination during the sample preparation and the analysis. For that, it is always recommended to have an ultra-clean lab with a plastic-free equipment and with a different type of strategies and solution in order to prevent any type of contamination. In terms of identifying gaps and possible future step in the research, we have, I may somehow recommend to assess a comprehensive assessment of the health risk posed to microplastic, because this is still lacking. The problem is so complex, because when we are discussing with ecotoxicologists, there is always this a question: is the plastic per se that is bringing toxicity, or is the chemicals that are released like a plasticizer, or chemicals that have been absorbed by the plastic during its journey into the environment, no matter if it is aquatic or terrestrial environment. So future research should prioritize some long-term health impact studies. Additionally, there is need for a better alignment between research findings and policy action, because research is going on a faster speed respect to the policymaker, and the policymaker need to be informed in the correct way with what type of action, what type of guideline we needed to have in order to prevent further pollution and suggest mitigation. Finally, the absence of a centralized database for microplastic research highlight there is the necessity for creating an international platform for data sharing and collaboration. This is true if, for example, we are comparing the American scientific world with the European one. We basically have a separate database. They are not interacting appropriately. This is like my initial kickoff of reflection.
All right, thank you, Alessio, for your opening remarks. And now our next panelist is Dr. Lee Thornton Hampton, who is a senior scientist at the Southern California Coastal Water Research Project's Toxicology Department. Leah?
Thank you so much for the introduction, Rachel, and for the opportunity to contribute to this panel. Before I introduce myself, I would like to introduce my agency, the Southern California Coastal Water Research Project, also known as SCORP. SCORP is an aquatic sciences research institute that works to improve the management of aquatic systems in Southern California and beyond. Since its founding as an intergovernmental public agency in 1969, SCORP has been developing strategies, tools, and technologies to more effectively protect and enhance the ecological health of SoCal's coastal ocean and the watersheds that drain to it. As an R&D agency, SCORP's reputation is built on conducting research and translating the science into actionable guidance and recommendations that inform the management, decision-making community, and policy-making development. I've been in the field in microplastics since I started my position at SCORP almost 5 years ago. During this time, I've had the opportunity to participate in collaborative international efforts to evaluate and standardize methods for collecting and analyzing samples for microplastics in a variety of matrices, including drinking water, stormwater flows, ambient water, sediment, and biological tissues. And I've also had the opportunity to help coordinate and contribute to international efforts to develop health-based thresholds for microplastics and work towards better estimating the risks that microplastics may pose, particularly to aquatic organisms. I also maintain the Toxicity of Microplastics Explorer, which some of you may have heard of, which is also known as TOMEX, which is an open source, open access database for microplastics toxicity data with an accompanying web application. During my time working on these efforts, I think there are many opportunities for filling out what I refer to as the microplastics monitoring toolbox and improving upon kind of the foundational existing tools and frameworks that we've started to ensure that these resources are accessible and usable by not only researchers, but environmental managers as well for large-scale monitoring efforts. In particular, there is ample opportunity, which many of the other speakers and panelists have highlighted, to generate more fit-for-purpose microplastics toxicity data so that studies are designed to meet minimum requirements so that we can better our first pass at threshold development for assessing risks and to get a better handle on doing risk assessment as well. Thank you.
Thank you so much, Lee, for your opening remarks. And now, Jean and Amari, if you could join Alessio and Lee, and we're going to begin our Q&A session with the full panel. So there's a number, and a reminder for the audience out there to submit your questions on the Slido platform, and you can also upvote for any of the questions if you don't have questions of your own. And so I think the first question that I want to cover for between all of you is, there's a lot of information out there on microplastics and a lot of research happening now and a lot of discussions across policy landscapes. So how can we increase public awareness of the issue of microplastics? And I don't know which of you want to take this first. How about we start with Alessio? So I know you were talking about communicating to policymakers and the public, so we'll start with you, Alessio.
Well, in my opinion, we first needed to bring our, scientific background. Not all the information are, in my opinion, at the moment mature. Especially when you are talking about the human health, we need to be a little bit precautionary in what we report, because this basically can also create a bad parallel communication. At the moment, in the research center that I'm collaborating, we also are targeting human samples, and listening a little bit also what the other speaker here have been also presenting. There is still uncertainty about the characterization and quantification. No matter the type of analytical equipment that you are using, we discussed about the pyrolysis before. Co-pyrolysis of material is also a problem there, which can also create artifacts during the characterization. So if you end up, providing such type of results, you may create some problems. So in a way, there are different mechanisms. Webinar like this one is a good mechanism to disseminate, definitely, for the broad audience, but also sitting and discussing in a thematic group, interdisciplinary one at a non-governmental or governmental level is also good. And of course, trust in the multidisciplinary approach, having on the same table different type of expertise, because you may bring your specific expertise as an analytical chemist or toxicologist, but you may neglect or not consider other opinion from social science and socio-economical people. All this is forming the message that the policymaker need to have.
Thank you, Alessio. And I want to let the audience know that this is a webinar series, and so we do have a webinar that is dedicated to risk communication. So I encourage you all to tune into that one when that is made public. But I want to move on to a question that was upvoted on and I guess we'll start with our initial panelists, Amari and Jean. Has there been any documented evidence on microplastics in soil being absorbed or taken up by agricultural crops grown in contaminated soils?
So when we're thinking about crops in particular, we have to think about what can cross through the cell wall, right? So if we're talking about microplastics in particular, they're a little too big in theory to be able to get into the roots or into the plant. But there are some studies that have been done using nanoplastics, mostly polystyrene nanospheres, that have shown some form of uptake into sometimes the leaves or the roots or into the fruit sometimes. So again, it's only usually a one type of particle, just spherical, but there is some evidence that there's the potential for others.
And Jean, did you want to touch on that?
Mari, you put that fantastically, so thank you. It's interesting too when we talk about impact. There have been studies that share not necessarily conflicting, but interesting results on the impact of nanoplastic uptake, whether it improves plant performance or decreases plant performance. So all of that is still in question. Yeah, we'll just emphasize that the size of the particles is extremely important. And often when looking at particle uptake, the focus is typically on root systems or more direct contact with the soil rather than seeing complete migration into other parts of the plant. And Leah, I see you nodding as well if you have any additional information.
Yeah, I think that both of your comments really highlight something I think about a lot, which is kind of the balancing act between the collection, the analytical methods, and the toxicology that collaboration across different specialties can really highlight. So you mentioned that size is obviously incredibly important to this type of exposure mechanism. And therefore, if this is the type of thing that you're looking for in a study, you need to make sure that the sizes that you're using in your, let's say, laboratory study are making sense, and that you're using the appropriate analytical methods to be able to fully quantify and characterize those sizes of particles. So
On that note, are there critical characteristics of microplastics that should be prioritized in risk assessment? So you mentioned size, but are there other things that we should be looking at as prioritization, whether that's shape or polymer composition or chemical additives, bioavailability?
Yeah, I'd say absolutely. From my perspective, the chemistry of the polymer is extremely important. Polystyrene, we've mentioned this multiple times. It's used primarily for toxicology studies because it's readily commercially available. It's easy to make those particles and they're well characterized. But the surface chemistry of polystyrene is completely different than some other polymers. And it's well known that surface chemistry can have an impact on how there is an interaction with cells, as an example. Size, shape is important. If you think about where just a particle of dust, for example, can end up in the respiratory system, the smaller that particle is, the deeper it can penetrate into the respiratory system and the lungs. And so I'd say by and large, the smaller end of microplastics are of interest for hazard and risk, but also size, shape, chemistry, lots of things.
Yeah, and I'd add even a lot of these particles don't usually have a lot of chemical additives, the standard materials that are made. So they're not as entirely relevant to what polymer materials you might see in the real world. So just, honesty or characterization of what kinds of additives that are placed in it, whether they're organic or inorganic in nature. And if we have quantitation of what's in there, that would also help, because it's such a complex idea that we've got these particle effects, but then we also have hundreds of different chemicals that can also be being released and causing different impacts too. So it's a very complex soup.
Yeah, and Amari, you had mentioned in your presentation the diversity of microplastics. And this is for the group. Are there specific considerations that are needed from an analytical standpoint to ensure that you get the data that can be interpreted properly?
I think about one thing that's happened in the last couple of years is just really great databases for when I'm thinking about the additive side of things. There's really great additive databases now that are accessible for groups if we're trying to do further chemical analysis using like high resolution mass spectrometry for environmental samples. But now there's starting to be more reference libraries, like we mentioned in the slides, for Raman and IR to be able to utilize. And I think one thing I didn't point out was that they're environmental spectral libraries, because there is a huge difference sometimes in the spectral readout of an aged polymer. So there are things that are being built to try and help us get on the same page for characterization.
Any other panelists have comments to that? All right, so so you all brought up materials, and I want to touch on that because microplastics are now facing the same type of methodological and regulatory challenges as nanomaterials, particularly in this area for standardized sampling, for analysis, for reporting, and all to ensure that we have data comparability and reliability. Are there lessons that you all are aware of from the nanomaterial space that can help address some of the critical gaps that we're seeing in microplastics characterization and risk assessment?
I think I'll just echo what Jean said earlier about size and surface chemistry. We know that these were critical when we were trying to figure out what was going on with nanomaterials. And there's already pretty good consensus that size is massively important for microplastics too. So I think that, oftentimes microplastics are talked about as, this incredibly complex suite of contaminants. It's so crazy. We've never seen anything like this before, but there's many contaminants that are hugely diverse, and nanomaterials is obviously the thing that's closest to it because you're dealing with the chemistry and the physical particle itself, but just kind of taking a really big step back and looking at, okay, when we had this challenge before with other classes of contaminants, how do we go about tackling this, and what can we take and borrow and apply to microplastics, rather than kind of throwing our hands up and being like, oh, completely overwhelmed, so there's just too much here.
Yeah, that's a great point, Leah. There's certainly a excellent set of frameworks across the globe for handling nanomaterials. I will throw a wrench into the works, of course, and say for most of the nanomaterials, we're focused on inorganic chemistries. And we know that we have particles at the nano level. For microplastics, when we get into nanoplastics, that transition period is not well understood. So when do we have transitions from solid particles to waxes to oligomers and single molecules? And if you think about, 1 nanometer particle, you have maybe 12 or less carbon-carbon bonds in a chain. So that's now a molecule, that's not a particle. There's a lot of questions when it comes to the physics. We know for microplastics, when we think about them in health assessment framework, that particle component is critical. But then what happens when we get to these smaller and smaller sizes? So that's just my, deep analytical and physical chemistry questions to throw in there.
All right.
Also,
I don't know, go ahead,
yeah.
Alessio.
Yeah, sorry. Also, perhaps we also need to stress that what we learn about the microplastic perhaps cannot be applied straight away to the nanoplastic, both in terms of collection, because it is really difficult to collect them in the environment. They
Right.
also follow specific, behavior. They aggregate. They also sink, so they are not really present as a nano, especially in the marine environment. And it is also difficult to set up like recoveries and get the proper analytical equipment for a correct characterization. Mass-based, you normally stress the instrument up to the minimum detection limit. I'm talking about the pyrolysis GCMS, for example. You need to load a lot of this nanomaterial in order to get the signal, for example. And the same goes with the vibrational microscopy oriented method. We also learned that they have like limitation. For the FTIR, micro FTIR, it's basically impossible to detect them. For the micro Raman, there is hope until 800, 600 nanometers, but nothing below. After that, you need to use a way more complicated type of equipment, not a lot of expertise available, and not really familiar with such type of sample. We definitely can learn more about, or at least learn from the lesson while analyzing nanoplastic, but again, it's not linear, the correlation. In a way, it's still like, pass me the word, a little bit a beast, and it is not really clear in a way if we are there in terms of characterization, and of course in terms of sample collection from the environment. The easiest matrix like drinking water and the marine water still give issues. Imagine when you are analyzing nanoplastic in sediments, sludge, or other very complex type of matrices.
Right. Well, what about for microplastics that are less than 20 micrometers? Are there recommended methods to collect these from surface waters like rivers and lakes? Those areas that are used for recreational purposes. If so, are they being standardized at the moment?
There is an ISO standard that has been released 1 year ago about the sample collection of water samples. And this is basically forming the basis.
Yeah.
Okay, and
For the ASTM standard, it's also using a 20 micron sieve. So it's not currently capturing anything smaller than 20. But I'll say I haven't seen anything, but if somebody has, send it to me because I'm curious.
What about, excuse me, recent studies they're using the GCMS to quantify microplastics in biological materials, but they don't evaluate potential interferences from incomplete sample preparation, such as fats. So how can this be addressed by the broader scientific community?
Yeah, I think that's a great question because pyrolysis GCMS spec itself is not a new technology. It's been used for decades to characterize polymeric systems. But having these complex matrix backgrounds is generally a really tough issue. One of the things that we're doing at ASTM is to start identifying what's the next frontier for standard methodologies associated with microplastics. We're going beyond the water and air matrices, and now what are the potential complications you have when you're utilizing biological samples? So certainly, I think webinars like this help spread the word. You know, interactions between scientists that are experts in pyrolysis GCMS spec will certainly help. And additionally, looking at secondary, tertiary confirmation for characterization. So not only using pyrolysis GCMS spec, but other techniques that can look beyond the matrix or, you know, serve as an additional QA/QC mechanism for confirmation or denial of results, depending on the situation.
Yeah, and Jean, you mentioned how to look at this progressively, how to think about the future. You know, what future methodologies do we need to think about to adapt to emerging concerns like nanomaterials, nanoplastics, and biodegradable plastics? So, I mean, from that standpoint, are there ISO and ASTM standards available for things? I mean, how adaptable are they? Are they developing fast enough for the needs of what we need to understand these very complicated issues?
Yeah, absolutely. And great question. So I'll speak from the ASTM side. Certainly, there are methods being developed now for more complicated matrices, but again, the focus has been on water. So we're not as concerned about potential interferences, matrix effects, so on and so forth. That exists for a handful of different techniques and does give us a good basis to stand on. There are additional methods being developed in ISO. Amari mentioned the compost extraction method, which provides sampling and analysis guidelines as well. But I'll hand it off to Alessio to talk more about ISO. And yes, we're moving forward, not as quickly as I would like, but that's just my personal opinion. It takes a long time to build, you know, scientific consensus. So completely understand why we're still working towards this, and hopefully we'll continue to grow leaps and bounds there. But Alessio, please.
Yeah, I mean, also I've been contributing in ISO standard. The development is a kind of a quite extensive process, and you need to get agreement with the different member committee across different countries. So it's not really an easy task. And finally, you know, the text is somehow what is in the agreement in the community, not necessarily reflecting the direction you would like to have. And it is not normally getting such type of detail, but at least it's a good starting point, you know, moving from harmonization to standardization. So to have everything done at the same way everywhere, because harmonization is going on the opposite way, in a way. So, and of course, targeting complex matrices, we needed to have like a better, good knowledge about what we are reading through, though. In some cases, at least for the microplastic level, we have been able, you know, to compare directly different analytical techniques and see linearity up to a specific level. In my personal experience, we tried to characterize plastic in drinking water across two different techniques, pyrolysis GCMS and macro FTIR. They were like incredibly linear up to 10 micron, and after that we basically lost the signal. It was not really stronger correlation. Another research group in Europe made the same for sediments. And they again found like a good correlation across the different type of technique. When it goes into the nanoplastic, I don't know if we'll be able to have some kind of complementary supporting technique, or at least the possibility to do combination of different type of instruments.
There's a question that came in from Slido on biological tissues. And so we had covered during the earlier presentations that there aren't standardized methods for this. Do you think that standards from other matrices could be leveraged to inform best practices, or do we need to redo all of the work that that's been done for each matrix? So for example, could we use some learnings from the accredited drinking water analysis method?
Yeah, great question. We can certainly use learnings from those methods, especially when it comes to general framework. But the hard part is that matrix, right? So we have different concerns for background levels of different molecules. And for every analytical technique, you have to evaluate what are the potential mechanisms for false positives and false negatives, and how do those background molecules that could be associated with microplastics impact those results. So you have to go through that process and that investigation all over again. And that's why it takes so long, right? If you're evaluating certain types of fats, polymeric materials, lipids, there's, these matrices are very complex. And so something that's developed for water versus for soil, you might not be able to use the same chemistry, the same oxidation mechanism, for example, to remove the biofilms found in soil versus, the lipids and the proteins that you have in blood. Very detailed analyses need to be, or very detailed evaluations need to be completed for biological samples.
Others on the panel want to address that.
Yeah, I just wanted to touch on something that Jean mentioned, which is something I think about a lot, is that I think everyone wants, for each matrix, like one true method. They do this. But I think the reality is that even within an individual matrix, you like sediment, you are going to have varying levels of organic material, different types of interferences. So I think that some of these standardization efforts and harmonization efforts that are happening right now are really exciting because they're recognizing that, and it's not a protocol that is like thou shall, do step 1, step 2. It gives you options and flexibility, and I think what's key about it is that they're built on, minimum performance criteria. So saying this is the target that you should hit for recovery of your particles in this size range and things like that. So I think that there's been a lot of progress, and the mindset going into this, I think, is offering a reasonable amount of flexibility while also ensuring that the data is high quality and reliable as well, because that's going to be essential for managers and decision makers. They need to know that data is rock solid before they make a massive investment or a decision one way or another.
Others want to hit on that?
Yeah, I'll just say that this is really complicated, especially when we're thinking about different matrices. But I think there's some things we can learn even from the sediment matrices that are, kind of being developed right now. The idea of density separations and then oxidations are probably going to be a key element in many ways throughout different types of matrices, biological matrices. But again, we want to preserve the microplastic at the end of the day. So we don't want to, deteriorate the polymer surface so we can't characterize it as well as we want with, things like FTIR. And we don't want to lose the material in the process for also things like pyrolysis. So it's a really delicate balance to try and remove organic matter and maintain the particle integrity. And that's why sometimes those experiments can last for hours to days to weeks to try and do this really slowly. And unfortunately, that's just the world of microplastics right now. It's time taking.
So we have a question that came in on the FTIR library search on microplastics, and the question is about the confidence on mixture analysis in the library search, because sometimes it doesn't yield one result. Instead, you have to perform a mixture analysis. So what is the confidence level on mixture analysis in the library search for the FTIR?
Oh, a challenge question. I don't think that I have any one recommendation from my end. But yeah, that's a good point. Things can get complicated quite quickly. I think the area that I wouldn't go into is look at very low confidence intervals. And of course, you have different mechanisms for having those matches as well, depending on your software. And the only advice I'm going to give here is use a secondary analytical technique if you're unsure for confirmation, and utilize reference materials that are as close as possible to what you believe you may see. But regarding a certain number, no particular advice from my end.
Perhaps I can add something on top of Jean. At least in my experience or in my lab, we built it up like a specific library for the FTIR, somehow adding also those polymers that were like collected from the environment, because normally, you know, the standard libraries are coming from standard polymers from the industry. But actually, with a different degree of oxidation, especially in the surface, you may have like conflicting results. So there was a kind of circular exercise. So analyzing first with FTIR, doing the analysis with the pyrolysis GCMS, and then going back. We, at least here, have like evolved quite a comprehensive library, which is also now also telling us a little bit also on the oxidation level of the plastic, which again could be like an additional information, may be useful if you want to track how much the plastic has been like in the environment, if it has been moved, if it has been again in collaboration with other specialists, analyzing, for example, the biofilm, try to understand the direction that this plastic has come or when has been collected by you and your specific equipment. So in terms of library, confidence may only increase if you actually train the equipment with everything that you have in terms of experience from different type of samples, different type of projects, different type of matrices.
Thank you, Alessio. And Lee, I don't know if you had a reaction to that question as well.
No, I don't have anything to add.
Okay, all right, no problem. All right, well, we are, I want to just lay out some of the key takeaways for this webinar, and that I saw from the conversation today and the presentations, and some of that was emphasizing the need for standardization there across all matrices for both biological and human sampling, and this is important because we need reproducible technique to improve data reliability and to ensure comparability across different studies. And so I think that is important. And the other key takeaway for me was the importance of transparency and quality in sampling and extraction and analysis. And this yields higher quality research. And the other thing that was mentioned here was, I think implicit to this panel discussion was the importance of interdisciplinary and cross-collaboration across a variety of different industries and sectors, and the need for harmonized approaches to advance some of the accuracy of microplastics monitoring and its implications that it has on human health and the environment. So I just want to thank the panelists for and the speakers for the work that you put in this webinar. We appreciate your time and your thoughts. I want to thank the audience for attending. And again, we are having future webinars in this, and I will now turn it over to Daryl for closing out the webinar.
Perfect. Thanks, Rachel. And thanks to the speakers and panelists for their thoughts. This has been a super productive discussion. But I also want to thank all of our participants who are watching. I just want you to know we had a lot of questions and we couldn't get to them all, but we will consider them all as we prepare for the next webinar in this series. Much like Rachel, I'm going to kind of step back here a little bit and summarize or try to summarize what we covered, which was a lot in this session. In the speaker session, what I heard was they highlighted the importance of appropriate sample, extraction, and analysis approaches. And I really love how they simplified it by breaking it down into those 3 consideration areas. So I think that's important for us to keep in mind. They also stress that different sampling methods have different complexities for different media, whether that be water, air, soil, or biological samples. When extracting samples, there can be matrix effects that can interfere with downstream analyses, including material identification. And of course, QA and QC are critical, including blanks, control and reference materials. And our speakers highlighted some best practices around method transparency and ISO principles. For the panel session, Rachel just did a great job of summarizing. I'm going to just maybe quote a few sound bites that I heard. Evaluation can be a balancing act between collection analysis and toxicology. We need collaboration to ensure good study design and interpretation. We need a centralized database of microplastics research for data sharing and collaboration. We need to learn from the past, leverage learnings from nanomaterial assessments to help inform characterization and risk evaluation approaches. And we heard a lot that standards are developing for characterization, but we need more from those ISO and ASTM standards. And I know people are working hard on that, and that's going to be a significant advancement in this space. And as you heard throughout, the one last thing I'll finish on that I heard was we need collaboration, collaboration, and collaboration, which is what I think is great about this webinar in terms of bringing different people and different perspectives together. So as we wrap up, this webinar was focused on sampling and characterization as a key foundational element for evaluation. The next webinar from this series will focus on the distribution of microplastics in the environment and then human exposure pathways. And that's actually going to be planned for March 20th. So mark that on your calendar and plan to attend. If you want to stay informed of these future webinars and EHMI activities, please subscribe to the Environmental Health Matters newsletter on our website. And finally, your voice matters. Take a few minutes to fill out the post-meeting questionnaire that's going to shape and inform future webinars in this series. So thanks again to all of our speakers, all of our participants. I really appreciate the dialogue and the active exchange. And with that, we'll adjourn the webinar. Have a good day, all.